Our DNA, the blueprint of life. It's a complex code that defines who we are at the most fundamental level. It dictates everything from the color of our eyes to our risk of developing certain diseases. This intricate code is responsible for the myriad of traits that make each of us unique. For decades, we believed this genetic code was fixed, unchangeable. Scientists thought our DNA was a static script written in stone. But what if I told you that wasn't the whole story? What if our genetic destiny isn't as set in stone as we once thought? Enter the world of epigenetics. This groundbreaking field is revealing that our genes are more dynamic than we ever imagined. This revolutionary field of study is changing how we understand the very nature of our genes. It's showing us that our genetic code is not the end of the story. Imagine our DNA as a vast library filled with books containing the instructions for life. Each book represents a gene, a chapter in the story of who we are. These books are our genes. They hold the instructions for everything our bodies do. Now imagine that some pages have bookmarks highlighting specific instructions. That's epigenetics at play. These bookmarks can turn certain genes on or off. It doesn't change the words in our books, but it tells our bodies which pages to read and which to ignore. This selective reading is crucial for our development and health. Our environment, our lifestyle choices, even our thoughts and experiences. They all have the power to influence these bookmarks, turning genes on or off without altering the underlying DNA sequence. This means our daily choices can have profound effects on our genetic expression. This means we have a surprising degree of control over our genetic destiny. By making conscious choices, we can influence how our genes behave. We are not just passive passengers on a predetermined genetic journey. We have the ability to shape our own genetic outcomes. We are active participants, holding the power to influence how our genes express themselves. This knowledge empowers us to take charge of our health and well-being in ways we never thought possible. Let's delve deeper into the intricate mechanisms behind this fascinating epigenetic orchestra. One of the key players in this complex symphony is a process called DNA methylation. Picture a light switch. When flipped on, the light shines brightly. When flipped off, the room plunges into darkness. DNA methylation operates in a similar manner. It involves attaching a tiny chemical tag, known as a methyl group, to our DNA. This chemical tag acts like a switch, effectively silencing the gene it's attached to, preventing it from being expressed. It's akin to placing a bookmark on a page, signaling the cell to skip over that particular gene. Think of it like this. You have a recipe book, but you only cook certain dishes based on your preferences and needs. DNA methylation is like marking specific recipes you don't use, ensuring those dishes are never made no matter what. This meticulous process plays a crucial role in regulating gene expression, ensuring that cells function correctly and efficiently. However, environmental factors such as pollution and diet can influence DNA methylation patterns, leading to changes in gene activity and potentially increasing the risk of diseases like cancer. This highlights the delicate balance our bodies maintain and the importance of understanding these epigenetic mechanisms. Another critical player in the epigenetic world is histone modification. Histones are proteins that act like spools, winding up our DNA into tightly packed structures called chromatin. Imagine a long thread DNA wrapped around numerous spools, histones. The way DNA is wrapped around these histones influences gene expression. Tight wrapping makes the DNA inaccessible, effectively silencing the genes. Loose wrapping, on the other hand, exposes the DNA, making it easier for the cellular machinery to read the genes and produce proteins. Histone modification involves adding or removing chemical tags to these histone proteins. These modifications act like bookmarks, signaling whether a particular stretch of DNA should be tightly wound or loosely coiled, thus affecting gene activity. Think of it like organizing your bookshelf. You might place frequently read books within easy reach while relegating seldom used ones to the back. Histone modifications work similarly, ensuring frequently accessed genes are readily available while keeping others tucked away. Section 4, You Are What You Eat, Diet and Gene Expression. We've all heard the adage, you are what you eat. This saying holds more truth than we might realize, especially when we delve into the world of epigenetics. Well, epigenetics shows us that this is more than just a catchy phrase. It reveals how our dietary choices can have profound effects on our genetic expression. Our diet plays a significant role in shaping our epigenome, the collection of all epigenetic marks on our DNA. These marks can turn genes on or off, influencing various biological processes. Certain nutrients act as methyl donors, providing the building blocks for DNA methylation. This process is crucial for regulating gene expression and maintaining cellular function. A diet rich in foods like leafy green vegetables, fruits, and whole grains can influence DNA methylation patterns, potentially protecting against diseases like cancer. These foods provide essential vitamins and minerals that support our genetic health. For example, studies have shown that a diet rich in folate, a B vitamin found in leafy greens, can alter DNA methylation patterns in genes associated with cancer development. This means that what we eat can directly impact our risk of developing certain diseases. Similarly, compounds found in green tea have been shown to have epigenetic effects, inhibiting the growth of cancer cells. These compounds can modify gene expression, providing a protective effect against cancer. Conversely, diets high in processed foods, sugar, and unhealthy fats can negatively impact our epigenome, increasing the risk of chronic diseases. These unhealthy dietary choices can disrupt normal gene function and lead to adverse health outcomes. These dietary choices can lead to changes in DNA methylation and histone modifications, ultimately influencing how our genes function. This highlights the importance of making healthy food choices to support our genetic health. It's like feeding our bodies junk food. It might taste good in the moment, but it ultimately harms our health in the long run. By understanding the connection between diet and gene expression, we can make more informed choices that promote long-term well-being. Section 5. Move your body, change your genes, exercise and epigenetics. Remember those epigenetic bookmarks? Well, it turns out that exercise is another powerful tool in our epigenetic arsenal. Physical activity can alter DNA methylation and histone modifications, leading to beneficial changes in gene expression, especially in muscle tissue. Studies have shown that exercise can increase the expression of genes involved in energy metabolism and insulin sensitivity, reducing the risk of type 2 diabetes. Moreover, regular physical activity can positively influence the epigenome of cancer-related genes, potentially lowering the risk of developing the disease. Exercise is like a reset button for our genes, promoting a healthier gene expression profile. So, next time you're debating skipping that workout, remember that you're not just choosing to be sedentary, you're directly influencing how your genes behave.